Hello, thanks for joining. We will begin in a moment. We want to give everyone a chance to log on. Hello, thanks for joining. We will begin our webinar in just a few moments. All right, Justine, let's go ahead and advance to the next slide. So hello, everyone. Thank you for joining us for tonight's webinar as we discuss an overview of gene therapy clinical trial studies for BAG3 dilated cardiomyopathy. My name is Nikki Butler, and I am the Director of Digital and Social Media for the Dilated Cardiomyopathy Foundation and the Genetic Cardiomyopathy Awareness Consortium. We're so glad you can be with us as we discuss this important opportunity. We have over 250 registrants for tonight's discussion. Next slide, please. Before we begin, I do have a few quick housekeeping notes. All attendee microphones are muted so that everyone can clearly hear our presenters. Please feel free to submit questions at any time using the Q&A feature in Zoom. We'll have a dedicated question and answer session following tonight's presentation. Please note that our presenters cannot provide individual medical advice. If you have questions about your own care, we encourage you to speak with your physician. Many individuals with genetic cardiomyopathy benefit from seeing a heart failure specialist at an academic medical center. We also ask that questions remain focused on the topic of tonight's presentation so that we can make the best use of our time together. And next slide. We do have a poll for tonight. We'd love to know who is attending tonight's webinar. So Justine, if you could push that forward, thank you. Are you best described as a cardiomyopathy patient, a family member or friend, a medical professional, or an industry professional? I'll give you a moment there to plug in your response, and thank you for doing so. All right. It looks like we have about 16% cardiomyopathy patients, 8% family members and friends, 25% medical professionals, and 50% industry professionals. So thank you. Share those. It is always interesting to see the breakdown on those results. So let's keep rolling. I want you to know that this webinar is being recorded and will be posted on our YouTube channel following tonight's discussion. As registrants, you will receive an email with a link to the recording. Our YouTube channel also houses a library of our previous webinar recordings, and using the auto-translate feature, these videos are available in 54 different languages. And next slide. So to begin, I'd like to give you just a little background. The DCM Foundation, focused on dilated cardiomyopathy, created the Genetic Cardiomyopathy Awareness Consortium, or GCAC, in 2023. This effort began in 2022 when the DCM Foundation recognized a major gap. Genetics in cardiomyopathy wasn't being widely discussed, not by medical professionals and certainly not by patients. And we realized something important. You can't talk about genetic cardiomyopathy in silos. Many gene mutations cross diagnostic boundaries. For example, a single gene variant might cause dilated cardiomyopathy, DCM, in one person, and arrhythmogenic cardiomyopathy, ACM, in another. Because of that overlap, we needed a broader, more inclusive approach. So GCAC was formed to raise awareness and provide education about genetics and cardiomyopathy across all types. And next slide. In just a few years, we've made meaningful progress. Awareness of genetics and cardiomyopathy has increased significantly, both in the medical community and among patients. When we launched GCAC, we started with 8 patient groups. Today, we've grown to 11, and we're continuing to expand. This momentum shows there's a real need and real interest in understanding the genetic side of cardiomyopathy. Next slide. These are the 11 patient groups that are participating in the Genetic Cardiomyopathy Awareness Consortium, along with the DCM Foundation. And we want to thank each group for participating. It's incredible to have a coalition of 11 different groups working together to solve a real problem with patients. And next slide. We're honored to welcome two incredible speakers with us this evening. First, Dr. Mayur Kasetty, Associate Vice President and Global Program Head for Cardiac AAV Gene Therapy at Rocket Pharmaceuticals. Dr. Kasetty is deeply involved in advancing gene therapy research for genetic cardiomyopathies, with a focus on developing new approaches that aim to address the underlying cause of disease. We're also pleased to welcome Dr. Barry Greenberg, a cardiologist and professor of medicine at UC San Diego, where he serves as director of the Advanced Heart Failure Treatment Program. Dr. Greenberg has spent decades caring for patients with heart failure and leading research to improve how these conditions are understood and treated. Together, they bring both cutting-edge research and deep clinical experience, and we're grateful to have them here to share insights that matter to patients and families. So now I will turn the presentation over to Dr. Kasetty. Thank you.
Great. Thank you so much, Nikki. And thank you to the DCM Foundation for providing this forum for us to share an overview of our gene therapy and natural history studies in BAG3 DCM. And thank you all for joining us. So next slide, please. What I'd like to do over the next few minutes is just give a quick overview of who we are at Rocket, how to think about clinical trials and gene therapy. And then I'll turn the mic to Dr. Greenberg, who will give an overview of BAG3 DCM and our BAG3 gene therapy study. And then I'll come back at the end to walk through our natural history study in BAG3 DCM, and then we'll have a Q&A session. Next slide, please. So at Rocket, our focus is on developing gene therapies for rare diseases. And what that means in practice is that we're trying to address the underlying cause of disease, not just therapies that manage symptoms. At Rocket, we spend a lot of time thinking about diseases like BAG3, where there's a very clear genetic driver and a real meaningful unmet need for patients. Next slide, please. So one thing that's really central to how we work at Rocket is that patients are the center of everything we do. And we really try to put that into action, not only in how we conduct our clinical studies, but also in outside projects and initiatives like access to genetic testing, raising awareness of rare diseases, and doing what we can to ensure that patients can actually access studies and therapy. I want to also really emphasize that the work we do does not happen in isolation. We work very closely with physicians and academic centers, patient advocacy groups like the DCM Foundation, and importantly, with patients and families themselves, whose perspectives are really the guiding light in what we do. We are constantly saying at Rocket that patients are our North Star. So next slide, please. So as I mentioned previously, we think a lot about rare genetic diseases, and we have programs in cardiovascular disease like BAG3, as well as PKP2 ACM, arrhythmogenic cardiomyopathy, and Danon disease, which is a form of hypertrophic cardiomyopathy. And I also want to mention that last month we received an accelerated approval from the FDA for a gene therapy in another rare disease program for leukocyte adhesion deficiency. And this really just reflects how the field is evolving and moving forward in a great way. So next slide, please. With that context, let's take a step back and just talk briefly about clinical trials. So next slide. So just very simply, clinical trials are how new therapies are studied in people. and they're designed to answer two core questions. First, is the therapy safe? And second, does it have the potential to provide benefit for future patients that have that disease or that condition? Before even getting to clinical trials, research starts in the laboratory, and then it typically moves to animal studies before moving on to clinical trials where they're tested in people. And clinical trials are entirely voluntary. These studies are highly regulated, and they're conducted with strict oversight to protect patient safety and the rights of participants. Next slide, please. So clinical trials typically happen in phases, and the early studies, what we call phase one, focus primarily on safety and have generally a small number of patients. Later studies, phase two or phase three or onward, start to look more at effectiveness and side effects and typically involve a larger number of patients. Some clinical studies will have additional phases beyond phase two to move to a regulatory approval with FDA or with other regulatory bodies. But this tends to be less common in rare disease research, where generally patient populations are smaller and there's higher unmet need for patients. And just generally in rare disease, the overall trial sizes tend to be smaller. But in general, the same standards around safety and rigor apply as they would for larger studies for more common diseases. Next slide, please. So now let's talk a little bit about gene therapy, because that's the approach that we're using for BAG3 and for our other genetic cardiomyopathies that we're trying to treat. At a very high level, gene therapy is about delivering genetic material that helps address the underlying issue that's causing disease. And an analogy I like is to imagine your DNA or your genome as a cookbook. And each gene is like a recipe in that cookbook that gives the instructions for a protein or a dish. In a disease like BAG3, the recipe that you might have has a small change where the final dish, which is the protein BAG3, doesn't actually turn out the way that it should to have its normal function in the cell. What gene therapy tries to do is deliver a new working copy of that recipe into the cell so that the cell can start making the correct version of the protein again. And that's kind of what this graphic on the right side of the slide tries to show. There are different approaches that gene therapy can take. It can introduce a new functional version of the gene. It can try to modulate the activity of an existing gene, or it can try to directly fix an error in a gene. And importantly, the approach that we use at Rocket for our cardiac gene therapies is to introduce the functional gene. It's like adding a new recipe into the cookbook. Next slide, please. So gene therapy, although it's relatively a new and innovative field, actually has quite a long history that really dates back all the way to the 1950s, where there's the initial proof of concept using a viral vector to treat cervical cancer. This gave way eventually to the 90s, where the first the first research in genetic diseases was conducted, all the way to 10 to 15 years ago, where we saw the first approvals, to today, where there are now dozens of gene therapies approved globally. As mentioned before, the most recent cell and gene therapy approval was actually for our own therapy. It's called Kresladi, and again, it's for severe leukocyte adhesion deficiency. And just overall, I think the key point here is that gene therapy is no longer theoretical. It's an active and rapidly advancing area of medicine that's really showing tremendous promise for hundreds, if not thousands, of patients. Next slide, please. So just to dive a little bit more into gene therapy and some of the questions that sometimes come up. So gene therapy does not replace all of your genes. It's really trying to target a specific gene And to use the cookbook analogy, it's not like we're rewriting the entire cookbook of 25,000 genes. We're just trying to give the cells in the body an extra working copy of the recipe or the right gene that it can then use to make the right protein. It's also important to note that gene therapy is not currently believed to change the genes that are passed on to children. So it wouldn't necessarily affect the inheritance of a genetic mutation. Patients' children's may still inherit their parents' genetic mutation. It's also worth noting that gene therapy itself is not currently known to impact fertility, although there are some gene therapies that are given with chemotherapy, and that chemotherapy can increase the risk of infertility. Next slide, please. So you may ask, how do we actually get the gene into the cell? To deliver the gene, we use something called an adeno-associated viral vector, or an AAV vector. This is a viral vector that has been studied for over 40 years, and it's particularly good at delivering genetic material into a range of cell types, such as heart tissue, muscle tissue, central nervous tissue, the liver, and several others. It's important to note that adeno-associated viruses cannot themselves replicate and are not by themselves infective. And these viral vectors have been engineered so that they don't have any ability to cause disease. They're really just there to deliver genetic information to the cells in target. Depending on the type of cell transduced, the material delivered by AAV vectors is thought to be able to persist in patients for the long term and potentially for the rest of their lives. And this is positive as that it may hopefully provide long-term benefit for patients. Next slide, please. So now we'll spend some time here because, like any therapy, there are risks to gene therapy. And the main risk with gene therapy is how the immune system responds to the viral vector. So the body recognizes the delivery vehicle, the AAV, as something foreign, and that triggers an immune reaction. What we typically see are things like elevations in liver enzymes, which tell us that the liver is under stress, and sometimes inflammation in muscle or other tissues. In rare cases, the immune system can become more activated and affect things like platelets, which help with clotting, and it can affect other organs like the kidney. You may hear terms with gene therapy like complement activation or TMA, which stands for thrombotic microangiopathy. And what these terms are referring to is an overactive immune system that's starting to affect normal bodily functions, including normal clotting and how organs normally function. These can be life-threatening when they are severe. This is why when we are administering a gene therapy to patients, we are carefully screening beforehand to make sure that patients are not at a higher risk for these complications. We also administer steroids and other medications that modulate the immune system. These are given both before and after treatment. And there is extremely close monitoring of patients, especially in the first 2 to 4 weeks after therapy, to make sure that these adverse events are not occurring and to try to intervene to mitigate them as best we can and as quickly as we can. Another important point to mention is that some patients will have pre-existing antibodies from prior exposure to similar viruses. So adeno-associated viruses exist out in the environment, and patients can be exposed to these viruses and develop antibodies against them. If those levels are too high, patients are not eligible for treatment because the therapy wouldn't work as intended, and it could actually increase their risk for a worse immune response. So overall, the risks of gene therapy, as with any therapy, exist, but they are very well understood, and we do have methods to actively manage them. But they are very real, And that's why early clinical trials are first and foremost focused on safety. Next slide, please. So with that overview, I'll now hand it over to Dr. Greenberg to walk through an overview of BAG3 and our interventional gene therapy study in more detail.
Great, thank you, Mayur, and my thanks to the consortium for the opportunity to talk to the audience here today. Let's go on to the next slide. What I'll try to do over the next couple of slides is give you an overview and an insight into BAG3 gene therapy, and this will also provide a little bit of an overview about dilated cardiomyopathy, because BAG3 is one of the forms of dilated cardiomyopathy, and I'll get to definitions of all of those during the course of the talk. Let me start out by just pointing out the magnitude of the problem, and that is that within the US, it's estimated there are about 30,000 individuals affected by BAG3 cardiomyopathy. Dilated cardiomyopathy is common in the United States. There are several million patients that go on and develop heart failure. About 50% of them are due to dilated cardiomyopathy, and about 40% of the dilated cardiomyopathy patients have an identifiable genetic cause. For the BAG3 mutation, which is an important cause of dilated cardiomyopathy, if you've got the gene, by the time you're age 40 years or above, there's about 80% likelihood that you will have cardiomyopathy. The gene becomes manifest in you. Overall, out of the patients with dilated cardiomyopathy, somewhere between 2 to 7% have this condition due to BAG3 genetic abnormality. So what is BAG3? BAG3 is a gene that encodes a critical enzyme. And when you're missing a copy of this, what will happen is that you'll develop abnormalities in cardiac structure and in function. The heart cells just won't be working normally. And what that means to the individual is that over time they will develop heart failure. That is the inability of the heart to deliver adequate amounts of blood to all the tissue in the body. Symptoms are fatigue, shortness of breath, fluid retention, cardiac arrhythmias. Let's go on to the next slide. So I talked a little bit about dilated cardiomyopathy, and let me get into a bit of a more detailed definition. And this is caused by the progressive thinning of the walls of the heart. So as that heart muscle begins to thin out, it impedes the ability of the heart to contract normally. And this ultimately leads to the development of heart failure and can lead to premature death. Patients will often experience symptoms at various stages of their lives and tremendous limitation in some cases in carrying out their activities of daily living, in being able to work, in even taking care of themselves. Now, dilated cardiomyopathy offers a therapeutic challenge, and the medical profession has really risen to this in developing a number of different therapies that are available. These include drugs, devices such as defibrillators, and even heart transplantation. And these are very effective ways of treating cardiomyopathy, but there are limitations. Not all patients respond to these therapy, and there's a number of patients that don't progress the way we'd like to, even getting the best of medical therapy. In addition, this medical therapy does not address the underlying cause of disease, so that although we may be able to treat these signs and symptoms and even improve cardiac function, the underlying disease is still present, particularly when there's a genetic disorder. Heart transplantation, although it's very, very useful, we do a number of them here at the University of California, San Diego, where I'm based, has its limitation because there are a number of short and long-term risks associated with cardiac transplant. And wherever possible, we'd like to seek alternative approaches for managing patients that have developed cardiomyopathy or disease of the heart muscle rather than having to replace the heart. Let's go on to the next slide. So this slide presents an overview of the study for treating patients with BAG3 mutation leading to dilated cardiomyopathy. Let's start down at the bottom and work our way up. And first, I'll point out that this is a phase 1 trial that is currently underway at our site and some other sites, and I'll show you a map around the United States pointing out where those sites are. And we're hoping to initiate this therapy over coming months in one of the patients at our medical center. Walking up in the slide now, if you look at the inclusion criteria, will involve patients who are between ages 18 and 65 with a clinical diagnosis of dilated cardiomyopathy and an identified pathogenic variant in the BAG3 gene. Patients are required to have a defibrillator in place for at least 3 months, and they also need to be symptomatic from their heart condition. We exclude patients if there's another etiology, another cause, if you will, of the cardiomyopathy, because you wouldn't want to be giving gene therapy for a specific gene mutation if there are other causes of why the heart isn't functioning normally. If patients are receiving intravenous medications and require to be sustained on those medications, that would invalidate them for the study. And then finally, if they have very severe heart failure or very, very low ejection fraction, that is a pumping measure of the heart's function, that would also exclude patients from the study. The trial is designed predominantly to look at safety, and we monitor patients extremely closely over the course of the trial, looking for any treatment emergent adverse events, particularly if these are severe events, or any dose-limiting toxicities related to therapy. We are also quite interested in getting efficacy evidence and efficacy signals. Predominantly, we're going to look for whether or not the gene gets delivered to the heart, whether or not it becomes functional there and it begins elaborating the protein that's deficient in the heart. And then we'll look at how that affects cardiac structure and function. And perhaps most importantly to me as a clinician, how does it make patients feel? Do they feel better? Can they exercise more? All of those are things that we're going to be looking at. Excuse me. Let's go on to the next slide. So if you would, a patient is found to be a candidate for the study, we take them through an intense period of screening where we go through their medical history and physical, and we talk with their referring physicians to get an idea about what their clinical course has been like. We get some of the old tests that have been done and the studies that have been done. And then patients who are deemed to be candidates for the study are invited to come to San Diego, where we repeat a number of those tests to get baseline values, which we're going to use to compare with after we treat patients with the gene therapy. This usually takes place over a period of a couple of days. And for those individuals who are then included in the study, we'll start pre-treatment with medications to tamp down the immune system response. And that will begin about 2 weeks before the initiation of gene therapy. For the actual therapy, we'll admit patients to the hospital and they'll stay in the hospital for a period of 7 days. And this is really to provide us the opportunity to have very intense surveillance of patients, measuring their heart function, measuring their heart rate, blood pressure, and to get daily labs and to have the clinical team interact with them on a very upfront and intimate basis. After about 7 days, patients are discharged. They remain in the San Diego area for a period of time, during which they come back and have frequent blood draws. We get tests periodically. And then about 2 to 3 months after the initiation of therapy, they go back to their home area, but we continue following them closely and we'll ask that they come back to San Diego at months 3, 6, 9, and 12, and also after 18 and 24 months after therapy is initiated. This gives us an opportunity to really get a very in-depth evaluation of the safety and the efficacy of the therapy that we're giving. Let's go on to the next slide. So as I mentioned before, we're currently open for enrolling patients now at the University of California in San Diego. There are two other sites that will be enrolling soon. One is under the direction of John Giudice at the Mayo Clinic in Minnesota. And the third site is under the direction of Dr. Dan Judge at the Medical University of South Carolina in Charleston. We'll go on to the next slide. And for additional information, there's a QR code that you can see on the slide here. And also contacting the website that's also listed. We can bring you up to date about where the study's going on and provide additional information about the clinical trials. And I'll finish up at this point, and we'll go on and talk a bit about the natural history studies.
Great. Thank you. Thank you very much, Barry. So I'll now spend a few minutes to talk about our natural history study, which is another important part of the overall program for BAG3. A natural history study is an observational study. And what that means is that patients are followed over time, but they are not receiving an experimental treatment. And the goal of these studies are to understand how the disease progresses, how quickly it changes, and how it varies between patients. And this is critical because it gives us a baseline. When we study a gene therapy, we need to understand what would have happened if a patient did not receive treatment. And this data is also used by regulators such as the FDA, the European Regulatory Agency, and others, as well as payers to evaluate how meaningful a treatment effect in a clinical trial actually is. Next slide, please. So what actually happens in these studies is that patients are followed over a preset period of time with periodic visits. And during these visits, we collect data from various assessments. They could be tests, questionnaires, blood samples, imaging, etc. And a key question that many people have is what happens to their data both during and after a natural history study. So that data is completely anonymized, and then it's combined with other subject participants And that combined data is then used to evaluate how the overall disease progresses. The combined and anonymized data can be shared with regulatory agencies and payers down the road to help compare disease progression against treatment outcomes for patients that have received therapy. But importantly, this data throughout the handling of it is completely anonymized. It is not directly identifiable to a specific patient. Next slide, please. So we are also conducting a natural history study for BAG3 in addition to our interventional study. And I just want to briefly review some of the key eligibility criteria for this study. So to be eligible, we are enrolling adults, so you need to be 18 years or older at the time of consent. You need to have a diagnosis of dilated cardiomyopathy as defined by mild to moderate systolic dysfunction. So that's mild to moderate reduction in heart function that's been recorded within the last 12 months prior to being enrolled in the study. Documentation of a pathogenic variant of the BAG3 in a CLIA certified lab. So this is a genetic test that shows that the variant of the BAG3 gene is actually one that can cause disease. And then NYHA class 1 to 3, which basically is a measure of overall function. And this would correspond to sort of that mild to moderate systolic dysfunction. There are also some key exclusions. And for example, previous participation in a gene therapy study, not a natural history study, but an actual interventional study where gene therapy was administered. Having another mutation in a gene that is not the BAG3 gene that may also be causing cardiovascular disease, and very severe or advanced disease, and then prior heart transplantation would be exclusions for this study. Next slide, please. So if you're interested in learning more, you can follow the QR code, which takes you to our clinicaltrials.gov posting for the study, or you can email clinicaltrials@rocketpharma.com. I do just want to mention also on this slide that we try to make participation as manageable as possible. So all travel, lodging, and related costs to get to the clinical site and all of the various testing are covered by Rocket. Next slide, please. So I'd like to close with a few brief takeaways before we go to Q&A. So first, we are on a mission to develop curative therapies for patients with devastating diseases. And we're striving to do that with gene therapy, which aims to address the root cause of genetic diseases like BAG3. Clinical trials are how we carefully evaluate the safety and potential benefit of these therapies. And natural history studies are a critical foundation that helps make those trials possible and that helps demonstrate that the benefits that we're seeing in clinical trials are actually real and that these therapies are going to be helpful for patients. I'll now hand it back over to the DCM Foundation team to lead us through Q&A. Thank you very much.
Thanks. Thanks so much, Dr. Greenberg and Dr. Kasetty. Really appreciate that great presentation, understanding BAG3 genetic therapies and your clinical trial and natural history study. Again, for all those attending, whether you're a patient, medical professional, or industry professional, we'd love to to get your questions and answer the questions if we can, which would be great. First question I've got is with the natural history study, do people need to actually go to the 3 clinical trial sites you mentioned to participate in it, or is it more virtual? I guess Dr. Cassetti maybe.
Yeah, sorry, Greg. Can you repeat that? So would someone need to participate in all like a phase 1 and a phase 2?
Yeah. So for the natural history study, because I know for the clinical trial, you've got to go there and get dosed and you test, right? So you're actually visiting the potentially three clinical trial sites, which is great. The natural history study, kind of how does that work? Do they have to go quite a bit? Is it mostly email online?
I
You know,
see.
okay,
Yeah, no, that's a great question. So we try to make the natural history study the least amount of burden for patients as possible, but we still are trying to collect data that's going to be relevant and help support our understanding of how the disease progresses. The way that we are currently structuring our BAG3 natural history study is to have yearly visits where a patient will come to a specific site. And so they'll come at their baseline visit and then at 12 months, 24 months, and we're following out to 48 months.
okay,
Patients are at their discretion can pull out of the study at any time. Of course, we do encourage that patients stay in the study once enrolled. But yeah, the visits are yearly, and as I mentioned before, we coordinate all of the logistics of going back and forth to the site. We're in the process right now of selecting sites across the United States and also in Europe. And so we're hoping to try to have a number of sites that are geographically distributed so that, the travel is less burdensome. Hopefully there's a site that's actually close by.
okay, great. And you did say, I think on the if I saw on the slides that travel for both the clinical trial and the natural history study expenses will be reimbursed because there could be a number of trips back and forth. So
Right.
it'd be great. Another question. Let me focus on the clinical trial if I could right now. And so if you have BAG3, you can be a patient anywhere. And let's just talk about the United States right now. Go to see Dr. Greenberg, which I know well, in beautiful San Diego, which is always a wonderful place to visit. So if I'm a patient, I could anywhere in the United States, I could select one of the three sites since they're up and decide which one to go to, right?
That is correct. We have patients literally from all over the country, actually from all over the world, coming to UCSD for gene therapy. And for the BAG3 study, throughout the United States, we can take patients from anywhere.
Okay, now that's great. And so there are also a number of medical professionals on, which I think are great. Hopefully, there might be some doctors, nurses, nurse practitioners, genetic counselors that might have some BAG3 patients. So they can send people anywhere. We have a QR, and they can actually send patients to you guys. We had a QR code. I think Nikki's going to present it again. That one, if you're a patient and are interested, there's a web page where you can apply and say, "Hey, I'd like to find out." Non-committal, but I can find out and get more information on this. And I also like to recommend if there's any medical professionals out there, if they would like to reach out to me about, hey, what are the next steps? I could put you, we've got to check with the IRB and all the rules, maybe put you in contact with Dr. Greenberg or some other people. But if your medical professional has BAG3 patients and want to have a conversation about what the next steps would be, I would say right now, just email me, gruf at dcmfoundation.org, gruf at dcmfoundation.org, or info at dcmfoundation.org, and we'll point you in the right direction to have the right conversations within all the rules. So you say patients all over the U.S. and world. We've got a couple questions of one, is it patients only open to folks residing in the U.S., or can it be Canada, which one question is, or other countries in the world?
Yeah,
So
when as Dr. Greenberg mentioned, it can literally be patients all over the world. Logistically it can be challenging in certain parts of the world. There's obviously travel and immigration and those types of challenges that we need to face. But with these types of clinical studies, yes, we're open to enrolling patients outside of the U.S., certainly Canada, and we have done this in some of our other studies. So this is really open to any patient that might be eligible for the study.
Okay, now that's great. And again, I know Minnesota's a beautiful, beautiful spot with Mayo, and obviously I know Dr. Dan Judge well, and in Charleston, South Carolina is a great spot. So yeah, that's wonderful. Um, Question, Amy, if you could. So there's a question on. Do we apply through the link or do our doctors need to apply for the study for us? That's a good question. Either Barry or Meyer.
Yeah, well, for all of the studies that we're doing now, I've interacted directly with patients who can reach out to me if they want to hear more about the study, or I've interacted initially with their physicians who have directed me to the patient. So either way works. But I'm certainly available, and I suspect the other investigators will be equally available to direct contact.
Okay, no, that's great. And again, if anybody wants to reach out to me, gruf@dcmfoundation.org, happy to, or info, I'm happy to have that conversation and steer you in the right direction. Um, Someone it says a horrendous issue with DCM and affected their family and it really horrific on their family, but they're in Scotland. And they're thinking that the travel might be difficult to participate, given that length, and I don't know their working situation and everything. What do you sort of recommend? I mean, Barry, you've done this a lot with people all over the world. How do you sort of answer that question?
Obviously, the further away, the more difficult logistically it becomes, because often it's not only the patient picking up and coming here for that initial 3-month period, but a family member accompanies them. And then we ask the individuals and the patients to come back every 3 months over that 2-year period. It is more difficult the further away you live. But it's certainly not impossible. And we've done patients as far away as Norway. So we
Wow.
can certainly overcome those. We try to make it as pleasant as possible. We do note a bit of an uptick in interest in coming here from the Midwest and Northeast during the winter months for whatever reason. But we try to make this as palatable as possible.
Yeah, no, that's great. Uh, I'm gonna go through some. We're getting quite a bit of. Uh, so why not include pediatric patients? Is a question that we have. Why just those 18 and over in this study, either study?
Yeah, this is a really good question. And this is something that we are sort of still discussing internally, whether we may expand the study to include pediatric patients or have a separate pediatric cohort to enroll. Initially, when we were designing the study, we wanted to reflect a very similar patient population that will be enrolled in the interventional study. So kind of as I was describing with natural history studies, these are really to generate information that's going to support our understanding of how the disease progresses and also to be used to sort of compare how the disease progresses versus how treated patients progress. And this is, sometimes used in regulatory discussions and for approval. That does not mean that we are ignoring the pediatric population. Very, very clearly want to state that we are sort of looking at potentially having a pediatric cohort or even expanding the age range. But that was just initially how we had designed the study because we wanted it to sort of mirror the patient population that was going to be in the treated arm.
Yeah, no, great. Another quick question that the slide had mentioned 30 to 70% of the people might be disqualified because of maybe antibodies. If for AAV9, which I believe is the therapy you're using, is it that high or,
No,
I've heard
no.
it's less than that, but
It's less than that. It's
Okay.
somewhere in the range of 10 to 20%.
Okay. Okay. No, that's great. And then we had a question about lamin A. Jim, I'm a lamin A patient. My children all have lamin A. There will be a clinical trial coming later this year that you'll hear about through us for specifically for lamin A patients. So just as an FYI on that one. How many in the clinical trial? You've got 3 sites across the U.S. How many people do you need in phase one of that for the phase one clinical trial?
So the phase one clinical trial probably will enroll somewhere between 6 to 12 patients. And
Okay.
it really depends on what we find from the patients, what we see as far as the safety and efficacy as this goes on. But it's a relatively small study and will involve just a limited number of patients.
Okay, great. Um, are you looking to enroll patients with a specific type of BAG3 variant, like deletion or missense? We obviously have an educated patient there. So yeah.
Yeah, obviously a very educated patient. So as long as it's one that causes it's a pathogenic mutation,
Okay.
it would not matter.
Real quick, and Maya, I think we've talked about, we have a friend that has the BAG3 P209L, which causes restrictive and a lot of neuromuscular issues. And one of the questions is, do you foresee these technologies being applied to patients with this deletion, with this gene, specific type of BAG3 gene? And you may not be able to answer that, but I wanted to ask that question.
Do you mean for extra cardiac abnormalities?
Yes.
Yeah, the answer is I don't know about that. We're really we'll be looking at a number of effects on different organ systems, but this really is focused on patients that have cardiac manifestations to begin with, and we'll be getting the tests that assess what impact this therapy has on the heart predominantly. So that may be something for the future, but the study currently is not focused on that population.
Okay. Thank you. A couple questions here. Is there a possibility that more sites will be added? Are you going to sort of stick, of the 3 in the US? And then the question about potential international sites.
Yeah, it is possible in the phase one that we may add additional sites. For in terms of Europe for phase one, we are just going to have US sites. But when we do progress to a phase two study, we will include Europe as well and potentially other regions in the world outside of the US and Europe.
Yeah. Okay, great. In your information presented, I noticed that the age range was 18 to 65. Is 65 the oldest included, or is there a little flexibility on that, at least from an upper limit of age?
Yeah,
65 is the upper limit at this point.
Okay.
Now that's in the phase one and phase two. We may expand that. Depends a lot on what we see during this. But for the phase one, those are the fixed ages for the study.
Okay. Okay. And do you see any, if for some reason it's hard to enroll or something, that there might be some flexibility on that? Because I think we have some people maybe just over that limit that are giving me some questions right now. So yeah.
So, yeah, we're always adaptable. That would require an amendment to the study protocol. Uh, but right now, we're sticking to those ages. Again, phase two may be different where there would be more flexibility right from the get-go.
Okay. No, great. That's great. I actually got a couple texts that some people are interested, so that's wonderful and very exciting actually. And of course, I won't say their name. Is there a discrepancy in a BAG3's variant pathogenicity? Oh, I can tell I don't not. How do you decide whether a patient is eligible for the study? So maybe kind of walk through that.
Yeah, no, that's a great question. And there are a number of the genetic variants in BAG3 that have been already identified as being pathogenic. We know they're associated with diseases. We know that they're causative. and any of those mutations will be candidates for the study.
Great. So you had mentioned also an ICD implanted for 3 months or more. Um, and is that for all patients in phase 1 or 2, or the first few? Does everybody need to have an ICD? Or if you could give me a little more information on that, that would be great.
Yeah, for the phase 1 study, particularly for the initial candidates in the study, an ICD is going to be a requirement. That's just a safety requirement for this patient population. Whether or not we maintain that in phase 2, again, we'll be learning a lot about the natural history of this disorder in parallel and from the clinical trial. And it's possible that in later phases of the study, that ICD requirement uh, may not be required for entry.
If somebody does not have an ICD but meets the other requirements, would you still like to have them reach out for later aspects of phase 1 or phase 2? So if I meet the other criteria, BAG3, pathogenic, all the other criteria, should I still sort of at least put my hat in the ring, so to speak, to be
Sure.
considered later on?
Firstly, I really do make myself open to talking with patients and their physicians, and would be glad to discuss this with patients who are interested in participating either now or in the future.
Yeah.
And keeping an open communication, I think, is important. Again, the criteria may change for phase 2, and there's also the natural history study that they can get involved in, which does not require the presence of an ICD.
Okay. No, great. And so I guess the idea is, hey, if you've got BAG3, maybe if you're not even exactly meet the criteria, it might be good to raise your hand. Nikki's going to, we've set up something with a patient advocacy organization that does this. So she's going to have, I think, a QR code after the Q&A is over with. And if are in the general criteria of meeting the criteria, maybe not exactly, maybe you don't have an ICD, maybe not, but you want to talk to an expert patient advocacy person, we'll give you that ability. We'll show you how to get a hold of that. Again, I think education is wonderful in these situations. So please reach out if you have BAG3 and just have questions, want to talk to somebody. Again, criteria may change at any time. That's all we have for questions for now. And I know we're running to time. Thank you so much participating. It's so exciting to actually have potential therapies. Nobody can say they're going to work or not, but we're so excited that there is potential hope for patients with genetic dilated cardiomyopathy and BAG3. So thank you guys for all the wonderful work that you do.
Yes,
No,
thank you
thank
all
you.
for being here, and thank you for taking the time to share and to learn more about this important research. If you're a patient interested in exploring the RP-A701 gene therapy in more detail, we encourage you to visit our webpage. There's a QR code here. It's a long URL. But you'll get an email with all of this in there as well. So on this webpage, you'll find clear, up-to-date information about the study, how it works, and what participation may involve. And next slide. You can also complete the inquiry form on that page. So yes, next slide. Yes. And if you complete this inquiry form, you'll have a trained independent patient advocacy expert reach out to answer your specific questions. And please know that filling out the form does not obligate you to participate. It's simply a supportive next step to help you stay informed and make the decision that's right for you. And next slide. So as stated earlier, registrants of this webinar will receive an email with a recording, and the email will also include a link to our specific landing page in the inquiry form, and also a link to register for our next webinar. So next slide. Our next webinar will be held Wednesday, May 20th at 6 p.m. Eastern Time. Our topic will be Understanding Wearables, Heart Data and the Okoro Heart Study. Registration is open and information can be found on the DCM Foundation and GCAC websites. And again, remember, even if you cannot attend live, just register and you'll receive a copy of the recording via email. Next slide. Thank you to all of our sponsors whose logos you see here on the screen. Their support allows us to present this important information. And thank you again to all of the patient partner groups for their ongoing support. Thank you, Justine, for your technical Zoom assistance behind the scenes. I much appreciate that. And final slide. Thank you all for joining us this evening and for being an important part of this conversation. We're especially grateful to Dr. Kasetty and Dr. Greenberg for sharing their expertise and helping us better understand the progress being made in BAG3 related DCM research and gene therapy. We hope this webinar provided helpful insight and a sense of direction as this work continues to evolve. If you have additional questions or would like to learn more, we encourage you to explore our resources and stay connected. Have a wonderful evening, be well, and we'll see you very soon.
Thank you, everyone. Thank you all.
Bye now, everyone.
Bye-bye.
Thank you.